Hi there, this is Dr. John Whitcomb doing a news and nutrition recording on peptides. And today we're going to talk about what peptides are and introduce you to the concept. You may not have heard about them before, so let's just be very basic and give you some key ideas here. Number one, a peptide is a small chain of protein made under 50 amino acids long. And some folks say maybe 100 amino acids. And you know, we're made out of 20 different amino acids, like 20 different colors of beads or 20 different letters of the alphabet. And so when you have 20 different letters, a protein chain that's only 50 of those letters long is a relatively small thing. Most proteins are can be in the range of several thousand amino acids long. In that context, they can make very precise shapes that fit things like antibodies and enzymes that do particular functions. So a peptide is a short little chain under 50. And actually you could call insulin the world's most original peptide. It's 51 amino acids long, so it's a little bit over the margin. In any case, if we look at your DNA, each cell in your body, each of the 37 trillion cells that make up you, have about 2 meters of DNA in them when you stretch it out. That's right. Your DNA would stretch all the way from the sun to Jupiter and back if you put all your cells end to end. 2 meters. Well, only 10 centimeters of that are the 22,000 proteins that make you uniquely you. What's the other 90%? Well, to date, we haven't known what the 90% is, and now we do. We now know that you code for something on the order of 300,000 microRNAs. Now, RNA is the mirror image of DNA, and it's the reflection of DNA, as you were, that can be interpreted by a protein called polymerase chain reaction that hooks up, that reads the code that's in DNA and turns it into a peptide and plugs the amino acids together in a chain reaction. And out the other end comes that peptide. Well, if you make 300,000 microRNAs, then you're making messaging for 300,000 peptides? Well, actually, all those RNAs aren't just coding for peptides. They're coding by themselves. They are also switches for DNA being turned on and off. And they're also, by themselves, their own little messengers. So there's huge complexity in your DNA. So the 90% of your DNA that is not just your proteins is the incredible complexity of what the human being is. It's your operating instructions. It's your iOS system if you're an Apple user, your Microsoft Windows if you're a PC user. It's your operating instructions on what your body should do when. Well, what we're realizing that at any given time, if we measure the exosome fraction of your blood, we can measure there are thousands of circulating peptides that are being sent out from one cell to another, either whether they be close or far, and that you have a whole cloud of circulating peptides telling your body what to do when. And then as we learn to read them, we can learn to manipulate them. And as we age, we also stop making them. So what you used to make in abundance as a young person, you no longer make as you get older. So what's happening right now is a little bit of like the California gold rush. 20,000 greedy prospectors with no sheriff in sight. There's an awful lot of excitement about peptides right now, but nobody's quite sure what to do with them. So we have this phenomenon going on of people trying out and finding quite interesting results as we use these peptides. So in the weeks ahead, I'm going to explore how these peptides work and introduce you to a bunch of these very useful ones. So hold your seatbelts on. Look, this is quite an adventure. We're going to teach you how the future of medicine is going to be shaped by reading your peptides. This is Dr. John Whitcomb with a News and Nutrition on Peptide Primer number 1. What's the magic of your DNA?